everyone, this is a video about protein synthesis by Mr. Reeks. Protein synthesis probably sounds a little bit dull, but actually it's really quite straightforward once you know the steps. The word synthesis might sound like it's a bit of a kind of sciencey word that you might not understand. It's actually really easy, it just means making stuff. So when you see protein synthesis, actually you know all it means is making proteins. And that's what I'm going to show you how your body does that today. How every cell in your body is constantly making all the proteins that it needs to do its job. And it does that using your DNA, your unique DNA. First, let's do a bit of a recap of what's in your cells. So, here we have a cell, looks like a fried egg, but actually here's the cell membrane around the outside. In the middle, we've got the nucleus. Just write that down here. Nucleus. And what I've tried to show in the middle of that cell is that it's absolutely full of these things called chromosomes. Human beings have 46 chromosomes in every cell, and they pair up actually to make 23 pairs of chromosomes. But actually we don't need to know about that for right now. What we do need to know is if you looked at any one of these chromosomes and kind of enlarged it and zoomed right in to find out what was inside, you'd see that it was really just one long strand of DNA looking kind of like this, kind of like a twisted ladder. And the structure of DNA that looks like this diagram here is really important. And I'm going to show you why. At the start of the point where the cell wants to make a new protein, it will go to one of these chromosomes, it will take one of these chromosomes, it will go to the DNA, and the bit of the DNA that it needs, it's going to unzip it, like so. It will unzip those two strands, so then you've got two strands of DNA that have now separated from each other, like that. And these two separate strands of DNA, you would be able to see, or you need to know that there are these things called bases, these particular chemicals. They're called adenine, guanine, cytosine, and thymine. But actually, you don't need to know that for right now. We use letters to describe them. A, C, G, T. And what you need to know is that T always pairs with an A. So if you have a T on this strand, it always joins with an A. And so when it actually makes the strand of DNA, they come together like that, and A always goes with T, C always goes with G. Looking at here, they split apart. Then now, as we say the DNA has unzipped, like so, it's actually now ready to do protein synthesis. What happens first is a copy of the DNA is made. Basically, inside your nucleus, it starts to make this new strand of something similar to DNA. It's called RNA, or in this case it's called mRNA. mRNA. And it stands for messenger RNA. And you'll see why in a moment. At the moment that's just a line, but actually what it does is that mRNA forms by making pairs with one of the existing DNA strands. So it'll make a T here to join with that A. It'll make a C here, a G here, because like I said earlier, G always goes with C, T always goes with A. That's just the way that the structure of DNA and RNA works. So this would be a G, this would be a and this would be a T. And then notice, actually, that this is identical to this strand here. And this strand of mRNA is now going to go off and do something different. The only other thing, for those of you aiming at A stars and higher, the only other thing to bear in mind is that mRNA is just slightly different to DNA in that it doesn't have the chemical thymine. It actually has something else called uracil instead. And so at A level and high level GCSE, you need to know that the T's are actually replaced with U's. But actually, in the terms of the way that works, it doesn't make any difference. The mRNA now leaves the nucleus, and I'm going to explain what happens in the next step now. Okay everybody, so I'm just about to describe what happens to the mRNA, which leaves the nucleus. Remember, the mRNA was that copy of the DNA, and in particular it was a copy of one gene of the DNA. So that means that it's a copy of just one of these little sections of DNA that happens to code for one protein. So this would be a gene, and the mRNA is a copy of that gene. The ribosome reads the mRNA 3 bases at a time. So it will read these 3 bases first, and it will know, there is just a universal code where each of these 3 bases tell the ribosome there is 1 amino acid that goes in this place here. That's quite a complicated process involving something called tRNA, but you won't learn about that until later on, so don't worry too much about that. Basically, the ribosome reads 3 bases at a time, and that is the code for which amino acid to put in that place. It reads along and moves down to the next 3, so the ribosome moves along from there to here, and it will read the next 3. Like this, those 3 G, C, U, and it knows that this is the amino acid that those 3 letters code for. And so that will make a big chain of amino acid as the ribosome moves further down and keeps going that way, reading along the mRNA. Obviously our mRNA here is only 6 bases long, but in real life it would be hundreds, even thousands of base pairs long. But the point is that the ribosome reads it by going from one side to the other and putting the right amino acid in the right place, depending on which of these 3 bases it's read. And so by the end, you get a lovely long polypeptide, which basically means a long chain of amino acids that eventually joins up to make one protein, this big long chain that folds over itself and makes one very large molecule, which is called a protein, and it can do all sorts of different jobs, as I've mentioned before. So that is protein synthesis. I'm now going to just do a quick explanation of what happens if one of your DNA bases changes. To really understand protein synthesis, you've got to explain how changing one DNA base can cause the entire protein that you're trying to make to be totally different. And actually, if you understand how it all works, you can already say that. Imagine that because of some random reason in one of your cells there's been a mutation, and so now this doesn't say C anymore, it actually says G, which means that this over here, this DNA, would actually be a C. And of course, the mRNA that's made is no longer a G here, it's also a C, because it's the only way to pair up with that G. Now suddenly, this mRNA doesn't say UAGGCU, it actually says UAC. So we've changed something, and actually that means that when the ribosome reads those 3, it's not going to put a triangle there, no siree, it's going to put something like a star. What I'm saying there is it will put a different amino acid in that place. And so if there's a different amino acid there, then when we flip this over and we put that into the place here, and this is no longer a triangle, it's actually a star, you end up with a totally different protein, because that star will change the way the whole thing folds up. And instead of looking like this, it'll look totally different, like this. And you end up with just a different protein. In fact, if you've changed one amino acid, you've probably ended up with a protein that doesn't do anything, because it's unlikely if you change the shape for it to actually do anything at all. You've probably just made a mistake in the protein. It now no longer has its shape. It now no longer does the job that it used to do. That's why DNA mutations are usually so harmful, because they end up with a protein not being made properly. And that, ladies and gentlemen, is protein synthesis. Wasn't that fun?